Okay, in the previous biochemistry video, we studied about carbohydrate chemistry, its classification, definition, examples for each class, monosaccharides, their classification depending upon the number of carbon atoms and the functional groups with examples as well. Now, in this video, we'll be studying about disaccharides. What are disaccharides? Its classification, its composition, and the three main uh, disaccharides which are important for your syllabus, which are sucrose, lactose, and maltose. We'll study about them in the coming slides. So, firstly, we'll see the introductory part of disaccharides. Disaccharide is among the oligosaccharide, and disaccharides are the most common form that are being used in our daily lives. So, as the name is evident, a disaccharide consists of two components of monosaccharide units, which are held together by glycosidic bond. Remember, there are two monosaccharide units which are held together by glycosidic bond. These disaccharides, their properties are crystalline, water soluble, and they are sweet in taste. So these are the properties of disaccharides. Now coming to the classification, disaccharides are of two types. One is reducing and the other one is non-reducing. Now what is reducing? A reducing disaccharide is the one which gives free aldehydes or keto groups, but a non-reducing disaccharides do not give any free aldehydes or keto groups. So, for an example for reducing disaccharide is maltose and lactose, and an example for non-reducing disaccharide is sucrose and trehalose. So now this gives the introductory part of disaccharides. Now we'll move on to individual disaccharides which are important for biochemistry. I mean the carbohydrate chemistry. So the very first disaccharide which we will be dealing with is maltose. A maltose is composed of two monosaccharides. One is alpha D glucose unit and the other one is also alpha D glucose unit. So there is a glycosidic bond between two alpha D glucose units. So these are held together by alpha 1 to 4 glycosidic bond. In the image I have represented the you can see in the second image there is a glycosidic bond between the C1 carbon of alpha D glucose and of C4 carbon of the second alpha D glucose unit. So there is a bond formation between C1 and C4 of two alpha D glucose units. Okay, so that is called maltose. Now we'll read out the points. Maltose is composed of a two alpha D glucose unit held together by alpha 1 to 4 glycosidic bond, and the free aldehyde group present on C1 of the second glucose answers the reducing reaction besides the Occasion formation, that is it forms a sunflower shape, and maltose can be hydrolyzed by diluting as dilute acids or enzyme maltase. So this maltose can be further divided or hydrolyzed by these dilute acids or the enzyme maltase, which gives two molecules of alpha D glucose. That is nothing but maltose on hydrolysis gives two molecules of alpha D glucose. So there are various types of maltose, which is isomaltose and celobiose. So what is isomaltose? That it contains the glucose units which are held together by alpha 1 to 6 glycosidic linkage. It has 1 to 6 glycosidic linkage, and celobiose is another disaccharide which is identical in structure with maltose, but there is a difference only in beta 1 to 4 glycosidic linkage. So celobiose is formed during the hydrolysis of cellulose. So these are the points under maltose. Read maltose again and again. they might ask what the composition of maltose or the structure of the maltose. So it is important for you to study maltose. Moving to the next type of disaccharide, which is sucrose. Sucrose is one of the sugar of commerce. That is, it is very much used in the market as well as in our day to day life. So this sugar is referred to as a cane sugar because it is being obtained from the sugarcane and the beetroots. So this sucrose is made up of alpha D glucose and beta D fructose. Note that sucrose is made up of alpha D glucose and beta D fructose. These two monosaccharides are held together by a glycosidic bond, which is alpha 1 to beta 2, repeating the glycosidic bond linkage, which is alpha 1 to beta 2 between the carbon atoms C1 of alpha glucose and C2 of beta fructose. Okay, so the linkage is between alpha D glucose and beta D fructose. So, in previous classification of disaccharide, we saw that sucrose was one of the non-reducing group. It was a non-reducing group because glucose and fructose are the two reducing groups which form a glycosidic bond. When two reducing groups of glucose and fructose are involved in the glycosidic bond, sucrose becomes a non-reducing sugar and it cannot form oxazone. It cannot form oxazone. So this is a non-reducing sugar. So this sucrose is a major carbohydrate which is produced by photosynthesis. It is transported from the storage organs of plants into roots, tubers, and seeds. And sucrose is most abundant among the most abundant naturally in naturally occurring sugars. So I have put a slide on uses of sucrose. So the first point says that sucrose is a major carbohydrate produced in photosynthesis, as I told, and it has a distinct advantage over other sugars as storage and transport form. It is transported into storage organs of plants, where like roots, tubers, and seeds. And moving to the next point, sucrose is the most abundant among the naturally occurring sugars, and sucrose is an important dietary carbohydrate also. So, sucrose is being added as a dietary component in our diet. Moving to the next use of sucrose, it is sweeter than most other common sugars, namely glucose, lactose, and maltose, with exception to fructose. And sucrose is employed as a sweetening agent in food industries. So sucrose is being used as a sweetening agent. Then the intestinal enzyme sucrase hydrolyzes sucrose to glucose and fructose, which are absorbed by the body. So these are the uses of the sucrose. uh, Moving to the next disaccharide, which is lactose, its composition and its uses. So, lactose is more commonly known as a milk sugar since it is being obtained from milk, and lactose is composed of beta D galactose and beta D glucose, which are held together by beta 1 to 4 glycosidic bond. So lactose is made up of two components, which is beta D galactose and beta D glucose, which are held together by beta 1 to 4 glycosidic linkage. So the anomeric carbon of C1 glucose is free. So hence the lactose exhibits reducing property and forms a oxazone, which is which can be observed by the powder puff or hedgehog shape. So the compound looks like a powder puff or a hedgehog shape. And lactose of milk is most important carbohydrate in the nutrition of young mammals. It is hydrolyzed by intestinal enzyme lactase to glucose and galactose. So this ends up the very important disaccharides that will be asked in your biochemistry exams. So I have put an image of lactose sucrose and maltose chemical structures. Please do refer to the chemical structures and remember the composition of it. What are the two glycosidic linkages, that the bond that is formed between the two components of disaccharides, two components of monosaccharides. So we'll just summarize the maltose, lactose, and sucrose structures. So maltose is composed of two alpha D glucose units. Remember that it is a bond formed between two alpha D glucose units, and the bond formation consists of alpha 1 to 4 glycosidic bond. Moving to the next one, which is sucrose, and sucrose is formed by alpha D glucose and beta D fructose, and the glycosidic bond is alpha 1 to beta 2 glycosidic bond. And moving to the last part, which is the lactose, and the bond lactose consists of beta D galactose and beta D glucose with the bond of beta 1 to 4 glycosidic linkage. I hope you understood the video. We'll be uploading some more videos on the chapter carbohydrate chemistry shortly. Subscribe to the channel to have update of the chapters that will be uploaded into the YouTube. Thank you.